Good evening, students. So in today's part of one shot, I plan to take heredity. So here, most of the chapters we already covered. So I thought like if we'll be completing this chapter, so this will be beneficial. Most of the students feel it to be boring. But in real sense, the chapter is quite easy. So here, very few things are there which you need to practice. So here, so we'll be waiting for a few minutes. Let few other students to join. Then we'll continue with the chapter. So here, this chapter, we are having more on you should have the chapter revision rather than going directly with the Vivek. I'll start in few minutes. Huh? So this for you only. I'll start with the screen sharing so that we can read the chapter fully. Will you say some questions where you feel difficulty in the chapter? Can you hear me clearly? Vivek, can you hear clearly? Can you hear? Vivek, is my voice clear? Not getting any response. Vivek, can you hear me clearly? So will you say like where you feel difficulty in this chapter? Topics which you feel very difficult. So here the in-text first question is very important. Triad exist in 10% of the population. In-text first question is very important out of this. What is heredity? Can you say answer? Okay, laws of inheritance. Total 3 laws are there, but in your syllabus you are having only one. Okay, DNA, chromosome, and chromatin. Okay. Chromosome already we had in 9th standard. That concept is enough for you to remember. Here they can ask about the full form of DNA and RNA. Okay, heredity is transfer of traits from one parent to another. Very good. So it's a process where the characters are transferred or the traits are transferred from parent to offsprings. For that transfer of the traits, which kind of reproduction is essential? Okay, let's starting the chapter now without wasting your time. So here, so as we plan for the complete chapter, once I'll be reading the chapter and explaining the things to you. We have seen that, so here, sexual reproduction. So here, so as we plan to complete out the chapter, so first I'll complete out the chapter. Most of the students skip this chapter. So I'll read it this in detail so that whatever the difficulties you are having, you can solve it. So here, we have seen that reproduction process give rise to new individual that are similar but subtly different. We have discussed how some amount of variation is produced even during asexual reproduction. Okay, during asexual reproduction also some variation may arise. Okay, generally we feel like during sexual reproduction only there is variation, but in asexual reproduction also sometimes variation arises. And The number of successful variations are maximized by the process of sexual reproduction. Variations are very less in asexual reproduction, as we know that in our sexual reproduction we get the true copies. So whatever the offsprings will be there, they will be true copies of the parents. Now in sexual reproduction there is variation. The main reason during sexual reproduction there is exchange of uh, chromatin material, like DNA exchange is there. Okay, in the ninth standard, we just touched the process of cell division. There are two kinds of cell divisions. One is called mitosis, and the second one is called meiosis. During mitosis cell division is carried in somatic cell. Somatic cell means the body cells, where the body cells will undergo development. But asexual reproduction is sexual reproduction during sexual reproduction, that is during meiosis, which are carried in the gametes. Okay, meiosis, there is a process which is called crossing over. There the chromosome material from both the parents, that is maternal and paternal, mother chromosome and father's chromosome, both get crossed over each other. And as a result of crossing over, there is exchange of genetic material, and that results in the variations. So that's why we get the maximum variations in sexual reproduction. Okay, so let's continue. If we observe, those who are having NCRT textbook, you can follow out from there. That will be much more easier to hear and follow out the sentences. If we observe a field of sugarcane, we find very little variation among the individual plants, but a number of animals, including human beings, which reproduce sexually, quite distinct variations are visible among different individuals. So when we are seeing the sugarcane field, it is by vegetative propagation. They cut out the stem and they plant it. So variations are not there, like very little variations, as it is producing by asexual mode of reproduction. But the other animals which reproduce sexually, there is a vast range of variations. In this chapter, we shall be studying the mechanism by which variations are created and inherited. So how the variations are created and how they are inherited, that is how they are passing from one generation to other generation. Now, accumulation of variations during reproduction. Accumulation means how they are getting accumulated, how they are getting increased. Inheritance from the previous generation provide both common basic body design and subtle change in it. So here, when the characters are inherited from previous generation, that when we are getting our characters from the parents, there is little, like body design will be same. Human baby will look alike like human beings only. There is change in the uh, other characters, like there may be change in the eye color or the hair pattern. So they will be the combination of both the parents. Now think about what would happen when this new generation in turn reproduce. So here the generations are going on reproducing. So there is intermixing of the characters. Characters are going intermixed. The second generation will have differences that they inherited from the first generation. So as sexual reproduction is going on, so there will be changes in the characters. There is the change in the genetic composition, as well as newly created differences. Now here we can see the figure. So creation of diversity over succeeding generations. The original organism at the top will give rise to, say, two individuals. So here it is like a single parent is there, which is getting divided into two offsprings. When we see the figure, the first one is completely blue, and out of that the second one, okay, some chapter name is Heredity. Okay, some of you are asking like which chapter is that? It's a heredity and evolution. Now, so there is change. We can see that there is a red color generation in the top, like head, or the base got changed with green color. So similar in the body design, but with the subtle difference. Each of them in turn will give rise to two individuals in the next generation, and each of the four individuals in the bottom row will be different from each other. So the variation is going on developing. The first one, the little change was there, and the second one, much more change was there. So here the changes are getting increased. So that's what we get in sexual reproduction. That is, the variations are getting increased. Now, do all the variation in the species have equal chance of survival? This question is very important. Okay, on the top I left few sentences. The four individual bacteria generated would be very similar. There would be only very minor differences between them generated due to small inaccuracy in DNA copying. So here the bacteria are being developed and they are developing by asexual mode. So here as the asexual mode is there, so variations are very little, and whatever the variations they are getting, that is due to inaccuracy in the DNA copying. However, if sexual reproduction is involved, even greater diversity will be generated. So during sexual reproduction, there is greater diversity. As we will see when we discuss the rules of inheritance. So further, we will read about what are the different rules. Do all these variations in the species have equal chances of surviving in the environment? This question is very important. All the variations don't have equal chances. Only the variation which can withstand the environmental factors, they only will survive. We can say that the dominant characters will be seen and the recessive characters will suppress. Obviously not. Depending on the nature of variation, different individual would have different kind of advantages. Bacteria that can withstand heat will survive better in the heat wave. So here whatever the character they can survive, whatever the character they can withstand, okay, that only they will enhance. So here if the bacteria is suddenly transferred into some hot climate. So here the bacteria which can survive the heat waves, they only will survive. Rest of all the bacteria will die. As we have discussed earlier, selection of variant by environmental factor forms the basis for evolutionary process. Now what is evolution? Evolution means how the organism got evolved. So here, as per the scientific research, we they consider like organism evolved by slow and steady changes in their features. Like as we know that human beings, they evolved from the apes. So here in the same way, like mm, as per the study, as per the history of the science, so they believe like the life first originated in the water, where some of the water bodies may started developing life due to hydrogen cyanide, which was present there at the time of formation of the earth. And some organism may maybe in the water, and suddenly they came onto the land. So they started developing the amphibian characters, which can be on the land and which can be in the water. So the first life started in the water. So first organisms which got developed were aquatic organisms. Then the later on, like amphibians, then reptiles, one by one, the different individuals got started developing. So how we can say that the evolution has taken place? Is there any proof? If we see ourselves, we are having certain proofs in our body itself, which explains that the evolution has taken place. In our body, appendix is there. Appendix was functional when the organism were taking the raw food. So here when the human beings were taking raw food, appendix used to produce certain enzyme which will digest the cellulose. So here now we are not depending on the raw food, we are having the cooked food. So that organ got reduced, but still it is present to give the evidence of evolution that once it was functional, but now it is vestigial. So we call all of them as vestigial organs. So in our body, we are having appendix as a vestigial organ, pinna is a vestigial organ, that is outer ear, and the next one, nictitating membrane. In our eye, at the corner, we have a pink color deposition like. So that pink color one is a nictitating membrane. When the human beings used to dive in the water for the search of food, so at that time, nictitating membrane was active. But later on, when they started avoiding that, So organ got reduced and its size got reduced on. So that still, but these are present in our body to give the evidences of evolution. So with all this, we can say that evolution has taken place. So no one were there who saw that before how the organisms were there and now how the advancement has taken place. Still, all these things, how they got concluded. So because of these evidences and the different rules or the different laws which were prepared by scientists. So there was a scientist called Augustus. He gave the law of use and disuse. So as per him, the organ which we use will develop and the organ which we stop using will reduce in size and it will remain in the body as a as a vestigial organ. It will reduce in size but it will remain, so that it will give a evidence of evolution. Okay, so to prove that, he took white mice and he cut the tail of the white mice for 21 generation. To his surprise, he found that even the 21st generation the mice was having tail. Then he concluded that tail is a organ which the mice need. If we cut it out, still it will have the capability to produce. Okay. In the same way, Mendel. So he was the father of heredity. He gave many rules of experiment like Mendel's monohybrid cross, dihybrid cross. So in this chapter, we are having all those which are very important. So out of the first page, you need to read this last sentence only. Do all the organisms. So this last sentence which we did. Do all these variations in the species have the equal chance of surviving? So if you are having textbook, you can mark out so that the revision can be done. Next one, after that, complete out all the in-text question and exercise question thoroughly. Now heredity, the most obvious outcome of reproductive process still remains the generation of individuals of similar design. The rule of heredity determines the process by which traits and characteristics are reliably inherited. So how the characters are inherited. Let us take a close look. So first one, inherited traits. So what are inherited? What exactly do we mean by similarities and differences? We know that a child bears all the basic feature of the human being. So by seeing the baby of a human, we can say that, so it is looking like a human baby as the similarities in the body design. However, it does not look exactly like the parent. The human population show a great deal of variations. So there are different variations. We can say that these are also, like sometimes they give the diagram and they ask about, there is a difference in the ear pattern. So some of the people have the earlobe completely closed, like completely it is attached with the skin, whereas the free earlobe. So here the free and the attached earlobes, the lowest part of the ear called the earlobe, it closely attached to the side of the head in some of us and not in the other. It is free. So these are the two different types of variations we see in the human beings. Now, rule of inheritance of traits. Mendel's contribution. So Mendel contributed by giving the inheritance rule, that how the characters are inherited from parents to offsprings. The rule for inheritance of such traits in human beings are related to the fact that both the father and mother contribute practically equal amount of genetic material to the child. So here, chromosome is the main, how the chromosome is being distributed. So nucleus contains chromatin material. Vivek, for you, I'm explaining this part. So listen carefully. When we see the nucleus before cell division, it is completely tangled. Completely, it is like a big network. So that network is called chromatin material. And during cell division, that chromatin material will uncoil and divided into small pieces. Those pieces are called chromosome. On the every chromosome is made up of two thread-like structures. So each individual one is called chromatin. Two chromatin joins to form chromosome. Two chromatids, they join at centromere. Okay, centromere is a place where two chromatids get joined, interlinked. Two chromatids interlink to form chromosome. On the chromosome, there is a deposition of DNA. DNA is made up of genetic material, and genetic material is nothing but it is a composition of protein, and that every protein has particular character. Okay, and that protein gives the character representation. So here, as per the Mendel, both mother and father are contributing equally. Like during this sex determination topic, we come across human chromosome where it is having 46 pairs, out of which 46 chromosomes, out of which 44 are autosomes and 2 are sex chromosomes. For male it is XX, XY, and for female it is XX type, and each one is providing one chromosome during the sexual reproduction. So here genetic material to the child. This means that each triad be influenced by both both paternal and maternal DNA. Thus, for each triad, there will be two versions in each child. What will be then the triad seen in the child be? So, child is getting from part from the mother also and father also. So, what will be seen? As per Mendel, dominant character is seen. Like in monohybrid cross, we saw that phenotypic ratio, genotypic ratio, the same thing will follow over here also. What is the dominant character? That will be represented. Mendel worked out the main rule of such inheritance, and it is interesting to look at some of his experiments from more than a century ago. So here Mendel did the monohybrid cross. So let's read about the monohybrid cross of Mendel. Mendel used a number of contrasting visible character. So he took the contrasting character, opposite character, like tall and short plant, white and violet flower, then round and wrinkled seed, then round and wrinkled seed. So these he took the contrasting character. He took pea plant with the different characteristics. A tall plant and a short plant produced progeny by crossing them. Progeny, that is offsprings, by crossing them, and calculated the percentage of tall and short progeny. So after having crossing, he calculated the percentage. In the first place, there are no halfway characteristics. So here in the first generation, what he saw that all were tall plants. We can see the diagram where the tall is crossed with short. Capital T are crossed with small t's. Where in the first generation, that is F1 generation, all capital T were obtained. So all T were obtained, that is all tall plants. Again, when he saw the genetic composition of this, that is when F1 is again crossed with F1. F1 generation, there is hybrid composition, that is one capital T and one small t. For this, I need to show on the paper. So later on, I will show you on the paper how the crossing is done. So here, capital T and small t, when crossed, he found that there is one completely capital T, two are hybrid T, that is capital T, small t, and one is completely short, that is small t. That is genetic ratio, genotypic. First one, phenotypic ratio. What is the phenotypic ratio? For this, phenotypic ratio is what we can see, 3:1, 3 tall, 1 dwarf. And genotypic ratio, that is 1:2:1. That is 1 pure tall, 2 hybrid tall, and 1 is pure short. So let's read in detail. No halfway characteristics, that is no intermixing. Either the complete tall was there, either complete short. No medium size was there. No medium height plants, all plants were tall. This means that only one of the parental triad was seen, not some mixture of the two. So the next question was, were the tall plants in F1 generation exactly the same as the tall plant? No, they are not exactly tall. Like they are having the hybrid composition. One triad of the completely tall and one is of completely short. So here, same as the tall plant of the parent generation. Answer is no. Mendelian experiment test this by getting both the parental plant and these F1 tall plants to reproduce by self-pollination. Then he did the F1 whatever the result he got for the F1, he made it to have the self-pollination again. The progeny of the parental plant are, of course, all tall. However, in the second generation, for the first generation, all were tall. Whereas in the F2 generation, progeny of the F1 tall are not all tall. Instead of that, 1/4 of them were short. So he got the 3:1. That is, 3 were tall, that is 75% tall plant, and 1/4, 25% were short. This indicates that both the tallness and shortness traits were inherited in the F1 generation. So in F1 generation, they were passed out, but only the tallness trait was expressed. When the F1 generation was seen phenotypically, on all plants were looking tall, but they were having the combination of both tallness and shortness, both the traits. So here the trait which expresses is called dominant trait, and the trait which does not express itself is called recessive trait. This indicates that both the tallness and shortness traits are inherited in the F1 generation, but only the tallness trait was expressed. This led Mendel to propose that two copies of the factors, that is, gene controlling traits, are present in sexually reproducing organism. So always two traits are present. These two may be identical or may be different. Identical, like completely TT, or it can be different, one capital T, one small t, depending on the percentage. A pattern of inheritance can be worked out with this assumption as shown. So here he calculated the pattern. In this explanation, both capital T and hybrid T, that is capital and small t, are tall plants, as capital T is the dominant one, while only if both are small t is short plant. In other words, a single copy of capital T is enough to make the plant tall, while both copies have to be small t for making the plant short. The trait like capital T is called dominant trait, while those which behave like small t is called recessive trait. Work out which trait would be considered dominant and which one recessive. So next figure they have given 8.4, where they took the purple one and green one. So purple flower and white flower. Okay, not green, purple and white flower. So here the purple flower is expressed more. So purple is the dominant one and white one is the recessive one. So this we completed. Next, what happened when P plants showing two different characteristics rather than just one are crossed with each other, okay, are crossbreed with each other? What do the progeny of the tall plant and a round sheet and a short plant and a wrinkle sheet? Now he took the two characters. Instead of tall and short, he took tall and round seed, and then short and wrinkle sheet, like two combinations. So they are all tall and have the round sheet. Tallness and round seeds are thus dominant traits. So when in the first one, F1 generation, what he found, all were tall and round. So he concluded that tallness and round is the dominant character and shortness and wrinkle is the recessive character. But in F2 progeny, again when it was self-crossed, then a Mendelian experiment will find that some F2 progeny are tall with round seed and some are short with wrinkle sheet. However, there would also be uh, um, some F2 progeny that showed both the combination, that is tall with wrinkle and short with round. So some of them would be tall but have the wrinkle sheet, while the other will be short and round sheet. So you can see as how a new combination of the triads are found in F2 offsprings when factor controlling for shade, shapes and seed color recombine to form zygote, leading to form F2 offspring. Thus the tall and short red and the round and wrinkle, they are independently inherited. So with this he concluded. So what he got, that when the crossing was done, some were having tall, and it's not like always the tall one will have the round seed only. So there was a combination. Tall were having some of the wrinkle seeds and the short one are having some of the round seeds. So with this, he concluded that the triads are independently inherited. So no intermixing is there. They're independent. Here only you need to practice out. This chapter seems to be very difficult, but when you will be learning, so learn out the in-text and exercise question as I'm directing you. The next one, sex determination. Mostly we get the direct questions, okay? Now, how do these traits get expressed? So how they are expressed? How do the mechanism of heredity work? Cellular DNA is the information source for making protein in the cell. This part is very important. Okay, DNA is responsible for making protein. A section of the DNA that provides information for one protein is called gene. Okay, the section or the part of the DNA which is giving information for particular uh, protein is called gene. How do protein control the characteristics that we are discussing here? So protein are controlling the characters like the tallness, shortness. So let us take the example of tallness as a characteristic. We know that plants have hormones that can trigger growth. So in the control and coordination, we read about auxin. Okay, auxin is responsible for the tallness of the plant. So plants have hormones which will trigger growth. Plant height can thus depend on the amount of particular plant hormone. The amount of plant hormone made will depend on the efficiency of the process of making it. So how the plant is controlling it? Okay, Ayush, once again you joined. Thank you for rejoining again. Today we started with bio section, which is, I think, not of your section. So here, okay, we were like hormones, how the hormones are under control. So yeah, particular hormone is produced by particular protein. Protein is responsible for producing hormone. So the amount of plant hormone made will depend on efficiency of the process of making it. Consider how an enzyme that is important for this process. If this enzyme works efficiently, suppose certain enzyme is responsible for producing certain protein. If that enzyme is not producing, that protein will not produce. If the protein is not producing, means it will not make the hormone to generate. If the hormone is not generating, it will control out the character, the adverse effect, like enzyme which is responsible for producing auxin. If it is not producing efficiently, auxin will not produce, which will trigger the growth of the plant, and plant will become short. Now, if this enzyme works efficiently, a lot of hormone will be made and the plant will be tall. If the gene for that enzyme has an alteration, so some defect is there, some alteration is there, and alteration that makes the enzyme less efficient. So enzyme is less efficient. The amount of hormone will be less and the plant will be short. Thus, gene control characteristics are tried. Underline this, Vivek. This point is very important. You need to learn like how genes control the characteristics or triad. We need to write the answer from upper paragraph by giving the explanation. Any difficulty, Vivek? Vivek, any difficulty? Not getting response, Vivek. Okay. So underline this. If you're having textbook with you, underline this paragraph. You need to learn properly. Now, if the in, interpretations of Mendelian experiments we have been discussing are correct, then both the parent must be contributing equally the DNA of the progeny during sexual reproduction. We have discussed this issue in previous chapter also during reproduction. Okay, so if both the parent can help determine the traits in the progeny, traits means the characters in the progeny, both parent must be contributing a copy. So as we know that I told just now, like a chromosome is made up of two chromatids, okay, and each parent is providing one chromatid. So each one is providing one copy of the same gene. This means that each pea plant must have two sets of all gene. So two sets like complete tallness or complete shortness, they are having two sets. One inherited from each parent. For this mechanism to work, each germ cell must have only one gene set. Germ cell. Ayush, yesterday you attended so nicely, you were answering. So again, I forgot you. And moreover, I'm having one of the student by your name. So he was from Barampur, Orissa, and he was a very good learner. He was a class topper. This name is very interrelated, heart touching to me. Okay, next one. If the, as per this interpretation, both the parents are providing one gene. For this mechanism to work, each germ cell. So here from the reproduction, we can have the germ cell. What are the germ cell? Male germ cell and female germ cell. So that can be one of the 1 mark question. Next, how do germ cells make a single set of genes from the normal two copies that all other cells in the body have? If progeny if the progeny plant inherited a single whole gene set from each parent, then the experiment explained in the figure 8.1 cannot work. So if the parent is providing complete single gene, like I told you, there is a crossing over where they will exchange the genetic material. It's not like the complete genetic material will be provided from one parent only. If only one will be providing everything, then the progeny will be either like the mother or like the father completely. They will not be having the combination of both. So here there is exchange of copies. It's not like the complete is provided by only one. So this is because the two characteristics capital R and Y would then be linked to each other and cannot be independently inherited. This is explained by the fact that each gene set is present not as a single long thread of DNA, but as separate independent pieces. Separate pieces, okay, small piece, like some of the part it will get from mother, some part it will be getting from the father. So there is exchange of copies, and whatever the segment they are getting from the parent, the combination they will have in the offsprings. So that we call genetic material which they are getting from the parent, that is separate independent pieces, and each one is called chromosome. Thus each cell will have two copies of each chromosome, one each from the male and female parent. Every germ cell will take one chromosome from each pair, and these may be either maternal or paternal origin. So they are getting the chromosome. For this, if you want to learn it in a better way, once you go through with search for the meiosis division and read it in detail. When we read the meiosis division, so there 4 steps are there: prophase, metaphase, anaphase, telophase. So like that, how the crossing is done, how the chromosomes are getting and divided, how they will arrange themselves, how they will cross out each other, then how the material is being exchanged. So if you see that part, you will get this explanation much more properly. Otherwise, you need to learn out. Only if you want to complete for your examination, keep in mind like each paternal, from both the parents, we are getting the copies. Then finally, they are being exchanged, and that will be in the offsprings. So here, when two germ cells combine, okay, when egg, that is ovum and sperm combine, they will restore the normal number of chromosome in the progeny. So here, what we can say, both the germ cell are providing, and if we count the number of chromosome, if mother and father chromosomes are getting combined, then offsprings must have the double number of copy. It is not happening. Why they're not having double number of chromosomes? So that's like during sexual reproduction, the chromosome will get divided into single set, which we call chromatid, and chromatid is passed over, and there is a interphase during the cell division. We call a phase which is interphase, and during that interphase, it will develop the next contradictory copy. Okay, whenever the DNA, this chromosome synthesis will take place, it will develop contradictory part, just opposite combinations. Before this, when old NCRT was there we were having the DNA structure fully. So there the complete explanation was given, how the contradictory pairs are there, what are the different proteins present, all those were in detail. So here you need to remember like only one single chromatid is being passed out, and that will develop its copy during the resting phase. So the number of chromosomes never get changed in the offsprings. They always remains same. Once again, I'm reading that sentence. When two germ cells combine, they will restore the normal number of chromosomes in the progeny, ensuring the stability of DNA. Okay, there is stability in the species. Such a mechanism of inheritance explains the result of Mendel's experiment and is used by all sexually reproducing organisms. That is, the number of chromosomes or the number of uh, mm, um, amount of DNA will be always restored. But asexually reproducing organism also follow similar rule of inheritance. Can we work out how their inheritance might work? Now the sex determination, already you did it. So we have discussed the idea that the two sexes participate in the sexually reproduction must be somewhat different from each other for a reason. How is the sex of a newborn is determined? From this paragraph, you need to remember out some points. Different species use different strategies for this. Okay, different species, how they have given like different species use this strategy. Like some depends on the environmental factor. If you recall back on that previous class, we discussed like some are related to the temperature. Temperature decides whether the developing baby will be male or female. Okay, some can exchange the sexes, which are called hermaphrodites, snail. So here some examples we'll be having, which you need to remember. Some rely entirely on the environment. Thus, in some animals, like few reptiles, the temperature at which fertilized egg are kept determines whether the animal developing in the egg will be male or female. Some reptiles, that is turtle. Okay, turtle, it depends on the temperature, whether it will be male or female. In other animals such as snail, individual can change its sex, indicating that sex is not genetically determined. So as per that, it's not fixed one. They can change it out. However, in human beings, sex of the individual is largely genetically determined. In other words, the genes inherited from our parents decides whether we will be boy or girl. But so far, we have assumed that similar gene set are inherited from both the parent. Okay, till now we were saying like both the parents are providing. But how the combination is there? Depending on that, whether it will be a baby boy or baby girl. We'll come to know. So let's move further. The explanation lies in the fact that all human chromosomes are not paired. Most human chromosomes have maternal and paternal copy. So maternal and paternal copy, one from the alligator and turtle, very good. So here, maternal and paternal copy, most human, somewhere it is. Most human chromosomes have the maternal and paternal copies. We have 22 such pairs, but one pair called the sex chromosome is odd. It is not always being a perfect pair. For women, it is X and X type, whereas the male, it is XY type. So during crossing, so this crossing you need to remember, like if the X combines with the X of the father, then there will be a baby girl. If the X of the male will combine with the Y of the female, then there will be a baby boy, XY combination. So with this crossing you need to आयुष, ये जो जय लक्ष्मी के नाम से ज्वाइन हो रहा है ना, वो एक्चुअली उसका मदर का आईडी है, उसका नाम विवेक है. So his name is Vivek. He is very interested and very attentive. रेगुलरली मेरा क्लास अटेंड करता है, और ये जो मैं ऑनलाइन क्लास लेती हूँ मोस्टली मैं उसी के लिए ले रही हूँ, उसके ही सिलेबस को रिवाइज करवाने के लिए. वो जो भी मुझे बोलता है ना, तो उसी के बेसिस पे मेरा चैप्टर नेक्स्ट मतलब जो क्लास है वो मेरा चलता है. ओके, नेक्स्ट वन. हाँ, यहाँ से अभी तुमको याद रखने का है, लाइक ये वाला क्वेश्चन सबसे इम्पोर्टेंट है, सेक्स डिटर्मिनेशन. नेक्स्ट वन, ये इंटेक्स्ट क्वेश्चन पढ़ लो. अच्छा चलो, आज जैसे हमारा टारगेट था, हमको ये चैप्टर 1 शॉट में कंप्लीट करना था, हमने चैप्टर कंप्लीट कर लिया है. हाँ, चैप्टर हमारा कंप्लीट हो गया है. नेक्स्ट विवेक, पिछला वाला जो हमने देखा था, अगर तुमको याद हो तो क्वेश्चन पेपर जो हमारा सैम्पल पेपर था, उसमें ये थर्ड क्वेश्चन आया था. अ मैन विद ब्लड ग्रुप ए मैरिज अ वुमन विद ब्लड ग्रुप ओ. सो ये क्वेश्चन एक काफ़ी बार रिपीट होता है. सेक्स डिटर्मिनेशन वाला क्वेश्चन एक रिपीट होता है. और नेक्स्ट यहाँ से फोर्थ क्वेश्चन एक्सरसाइज में से, हाउ इज इक्वल जेनेटिक कंट्रीब्यूशन ऑफ मेल एंड फीमेल पेरेंट एंश्योर्ड इन द प्रोजेनी. फिर उसके बाद मैंडलिफ का मोनोहाइब्रिड क्रॉस, डाईहाइब्रिड क्रॉस। और तुमने मेरे को लॉ ऑफ इनहेरिटेंस बोला था, तो मैं वो लॉ हम लोगों को चैप्टर में नहीं दिया हुआ है, स्टडी मटेरियल में है, तो उन लॉस का मैं पिक्चर लेके तुमको भेज दूँगी, ठीक है? फ़ोटो में सेंड कर दूँगी, तो तुम उसमें से पढ़ लेना। तीनों लॉ हैं, तो तीनों लॉ का जो है वो मैं समझा दूँ, मतलब तुमको भेज दूँगी। अगर हुआ तो मैं फिर शाम को तुमको वीडियो कॉल करके एक्सप्लेन कर लूँगी, हाँ? ठीक है, चलो हमारा चैप्टर कंप्लीट हो गया, दैट टू ऑन टाइम। सो टुडेज क्लास वाज वेरी स्मॉल, बट स्टिल वी कंप्लीटेड दिस चैप्टर। हाँ, नेक्स्ट विवेक अभी बताओ कौन सा करना है हम लोगों को नेक्स्ट कल के लिए, तो उसके हिसाब से मैं टाइमिंग्स का सोच लूँ कि कौन सा टाइम पे ले लें हम। कल का मेरे को मैसेज कर देना कौन सा चैप्टर लेना है। ठीक है, अभी क्लास एंड कर दे रही हूँ। आयुष, थैंक यू। आज भी तुमने क्लास अटेंड किया, दो द टॉपिक इज़ नॉट रिलेटेड टू यू, बट हैविंग अ गुड कन्वर्सेशन। नेक्स्ट, मैंने सब्सक्राइबर का लिस्ट चेक किया है, तुमने अभी तक मेरे चैनल को सब्सक्राइब नहीं किया है, सब्सक्राइब कर लो भाई। क्लास एंड करने वाली हूँ, कुछ कमेंट करने वाले हैं आयुष, या तुमने लीव कर लिया क्लास? विवेक तब तक बता दो कल कौन सा चैप्टर लेना है। आयुष, आर यू देयर स्टिल? अवर एनवायरमेंट, ठीक है, तो कल हम अवर एनवायरमेंट कर लें, पर आज के तुम ये आज तो ये श्योरिटी रखना कि तुम सारे इंटेक्स्ट और एक्सरसाइज के क्वेश्चंस कर लोगे अलॉन्ग विद दैट सेक्स डिटर्मिनेशन। हो तो मम्मी को ये चैप्टर का पूरा आंसर्स तुम करके दे देना, सुना देना उनको, ताकि ये चैप्टर तुम्हारा कंप्लीट हो गया ऐसा रहे। ठीक है, चलो क्लास एंड कर लो।